Good morning, everyone. If you're looking for Lunch and Learn by Clean Coast, Texas, you are in the right place. My name is Adriana Mendez. I'm the coastal coordinator with the Meadow Center for Water and the Environment. I'm here to facilitate this presentation. Jason Pinchback and Brandy Santi with the General Land Office will be moderating and helping with questions at the end. We ask you kindly to keep yourselves muted during the presentation. Your camera, you might leave it on or off. That's entirely up to you. Please, if you have questions or comments, make sure you share them in the chat box. And at the end, we will be happy to answer them. Also, keep an eye. I will be sharing useful links in the chat for everyone to check and have more information about today's topic. Today's speaker is Andy Myers. He's going to talk to us about oysters and water quality. We're sure you're going to enjoy this talk. For those of you that just joined, this is Lunch and Learn by Clean Coast, Texas. We're very happy to see you today. In a little bit, we're going to share a poll. We would like to know where you're joining us from and what is your association as a stakeholder. Please feel free to comment in the chat box as well. Well, good morning. It is 11 a.m. Welcome to Clean Coast, Texas Lunch and Learn. We have had many responses and we have many people join. So let's see who's here today. We have about 30% of our attendants are city or county staff members, 20% belong to nonprofit education or academic organizations, 27% are planners, engineers, or other professionals. 10% are coastal residents, landowners, business owners, and 13% identify as others. So please, if you feel like it, type on the chat box, what is your role as a stakeholder? We would love to hear from you. Okay, Jason.
Thank you, Adriana. Good morning. My name is Jason Pinchback. I work at the Texas General Land Office in the Coastal Resources Department. Thank you all for choosing to spend a little bit of your day with us here to learn about shellfish and water quality along our coastline. These lunch and learns were designed to be a simple, easy, and low barrier to entry, if you will, to have conversations, interactions with experts in different fields who are doing work along the coastal zone, looking at water quality and non-point source pollution. This effort is a part of the larger program of Clean Coast, Texas, which is working with different jurisdictions to enhance the management of non-point source pollution. Well, what does that look like? Well, there's really no one simple and easy recipe for that, but it starts with knowledge, partnerships, trust, and that's what we're working on right now. Without any further ado, I would like to introduce Mr. Andy Myers. Andy is an Environmental Protection Specialist 5 with the Texas Department of State Health Services Seafood and Aquatic Life Branch. Andy graduated from Texas State University with a Bachelor's of Science in Aquatic Biology. While at Texas State, he also spent 3 years as an intern with Texas Parks and Wildlife Department River Studies Program. He has been with Seafood and Aquatic Life Branch for 10 years. We're really excited to have him with us today to share yet another angle on water quality and importance, not just to our own health, but also the health of our ecosystems and our economy. So without any further ado, I'd like to welcome Mr. Andy Myers.
Thanks for having me. Honored to be asked to present. Look forward to sharing this information. Hopefully it's not too intense. You can get very technical when you're looking at calculations and some of the work that we do, but try to keep it kind of light and keep the flow going. Can you see my slides?
It's not showing up yet.
All right. How are we looking?
There we go. We're in business now. Thank you, Andy.
All righty. So thank you for the introduction. We'll jump right in. Basically, I'm here to present why the health department needs boats. It's kind of a joke. We get asked quite often when we're in the field, what do you guys do with those boats? What are you checking the health of the fish, checking the water? Sometimes we can say yes to all those questions. But essentially, we regulate the shellfish growing areas, and we utilize water quality studies to help guide us through the process. So our mission statement is to protect consumers from contaminants, diseases, or other health hazards transmissible or found in fish and shellfish. And through doing that, we perform the following functions. We do certification of molluscan and shellfish shippers, licensed crab meat processors in the state of Texas. We approve imported crab meat. We issue fish consumption advisories and bans in public waters. And we issue the designation of shellfish harvesting areas. So with the fish consumption advisories, we actually go through the entire process. We collect the samples ourselves, process them, send them to a lab for analysis, take the concentrations of the specific contaminants that we get back, and if merited, we issue fish consumption advisories or bans. The Seafood and Aquatic Life Branch Oyster Program operates under the collaborative guidance of the National Shellfish Sanitation Program and utilizes the NSSP model ordinance with the Food and U.S. Food and Drug Administration oversight. An important function of the program, the first picture you see here is we do some summer vessel boardings to verify time to temperature procedures are adhered to for our Vibrio management plans on the harvesting vessels themselves. Our certification program performs inspections of shellfish and Texas crab processors, as you can see in the other picture. The Texas Administrative Code adopts the NSSP model ordinance into code for the state. The model ordinance is published in the Guide for Control of Molluscan Shellfish. Further, Seafood and Aquatic Life Branch is charged with the responsibilities of administering the Shellfish Sanitation Program for the state of Texas as prescribed under Chapter 436 of the Texas Health and Safety Code. The Seafood and Aquatic Life Branch work collaboratively with Texas Parks and Wildlife, who is tasked with licensing of harvesters, patrolling shellfish harvesting waters, harvester education, and shellfish enforcement activities in the growing areas themselves. So the classification program, we develop sanitary surveys, open and close areas due to harmful events or potential pollution events, and we publish harvest classification maps. Some of the stats for the program, 1,765 square miles of bays are classified in Texas. 33 shellfish harvest areas are included in that. 337 active bacteriological water sampling stations are located in the coast. And we average over 2,300 bay water samples collected per year. Some important definitions. The differentiation between a growing area and a harvest area is very important to how we classify areas. A growing area is defined as any site which supports or could support propagation of shell stock. Shell stock is living molluscan shellfish, and that could be by natural or artificial means. A harvest area is an area that contains commercial quantities of shell stock and may include aquaculture sites and facilities. So sanitary surveys have certain requirements. The principal components of a sanitary survey include an evaluation of the pollution sources that may affect areas, an evaluation of meteorological factors, a review of hydrographic factors that may affect distribution of pollutants throughout the area, and an assessment of water quality. In doing so, we describe the growing area, document actual and potential pollution sources, evaluate all the characteristics that can affect an area. We conduct water quality studies, we interpret the data, and then we produce the classification maps. So there's three types of sanitary surveys: a comprehensive, triennial, reevaluation of the comprehensive, and an annual update. The comprehensive is conducted at minimum every 12 years per the NSSP model ordinance. However, in Texas, we complete these for each growing area every 6 years. Shoreline surveys are conducted to document both point and non-point sources of pollution. Some of the point sources we are interested in are municipal and industrial waste discharges, package treatment units, and potentially malfunctioning septic tanks. Non-point sources are rainfall events or stormwater runoff, agricultural and wildlife area runoff. We also generate maps from the comprehensive based off of the analysis and shoreline surveys. The triennial is kind of a reevaluation of that comprehensive survey. They're done every 3 years. We also do in-field shoreline surveys. We look at minimum 3 years of baywater bacteriological data. We reevaluate classification maps for any potential changes and reevaluate the overall comprehensive survey. Annual updates are done annually. We review shoreline surveys and look for potential changes. We verify that our sample frequency meets the NSSP requirements. We review 3 years of data, potentially up to 6. Review harvest area data in the open status and make classification recommendations. The model ordinance chapter 4 at 02, the authority in this instance would be DSHS, is tasked with establishing sampling strategies, and we utilize adverse pollution condition sampling and use fecal coliform as our indicator organism. Utilizing the 337 sample stations, sample typically well above the NSSP minimums. Coliforms are bacteria that live in the intestines of warm-blooded animals, so like humans, pets, farm animals, and wildlife. Fecal coliform bacteria kind of coliform associated with human or animal waste and indicate the potential presence of other bacterial and viral pathogens. So we utilize the fecal coliform as our indicator species, and they are described as gram-negative and non-spore forming rods. E. coli is included in this group of coliforms. It also, the chapter 4 we have to establish bacteriological sampling stations and at a frequency of collection which must be adequate to effectively evaluate all pollution sources, and also establish classification criteria for shellfish growing areas. So adverse pollution condition is a state or situation caused by meteorological, hydrological, or seasonal events or point source discharges that have historically resulted in elevated fecal coliform levels in a particular growing area. Image here was an image from NASA following Harvey. Really shows impacts to Galveston Bay. You can really see the lines, the freshwater flow carrying silt, likely carrying bacteria, any kind of other runoff. So DSHS utilizes a fecal coliform standard modified A1 method, which is a 5-tube dilution NPN test. Essentially, aliquots of bay water are diluted to 3 concentrations with 5 vial series. The number of positive vials for each dilution is compared to an NPN index to generate a fecal coliform most probable number per 100 milliliters. It's an accepted methodology. We get the results within 48 hours of sampling. Lab standards and the facilities are certified both by FDA as well as DSHS, and we have 2 labs we utilize in the state, which is our own DSHS laboratory in Austin and the Corpus Christi Nueces County lab. So how do we collect the actual bay water samples? Water samples are collected from a depth of 2 feet using a PVC sampling wand, which has a slot for fluid entry and a threaded end to attach a 125 milliliter polypropylene autoclavable and leak-proof bottle. Immediately after collection, the samples are placed in an ice chest surrounded by crushed ice, then either hand delivered or shipped overnight to either the Austin lab or the Nueces County lab. We utilize this testing because at the 2-foot level, you're capturing the effects of a potential meteorological event. Your salt water or more saline water sinks to the bottom. Fresh water flows over the top in a rainfall event. So you're gonna see the bacteria more likely there. Our Texas bays are relatively shallow, and so they mix pretty well. And it is a standard process that we can uniformly conduct through all 3 of our field offices. We have an office in Bay Cliff, Port Lavaca, and Corpus Christi. So the minimum requirements for the APC collection for approved areas, it requires 5 samples under adverse conditions. That's per year, and that's a NSSP requirement. DSHS tends to do more so, and we do it at least once a month. Oftentimes it's more than a single sample run per month. And this is for every approved area. For conditionally approved areas, it's 1 sample per month when the area is open. And also our minimum is once per month, regardless if it's open or closed. So adverse pollution condition sampling targets sampling that has historically held the highest fecal coliform levels, the window of December through February, which also coincides with Texas Parks and Wildlife regular oyster season. We tend to see more rainfall events associated with cold fronts. It reflects bacteriological water quality based upon current rainfall or river stage, as well as tidal movements. Sample frequency can increase if meteorological hydrologic events are extended. So in a scenario that we see a large magnitude rainfall or flooding related to river discharge, we'll collect samples multiple times throughout the month. Texas is unique in that we have a barrier island system, which kind of controls some of our hydrological movement. They're established and natural passes that funnel water in and out of the Gulf. We have a relatively low tidal volume as compared to the Northeast or Northwest, where they see, potentially 8-foot swings. We may see 2-foot. Our currents are primarily wind driven. Typically, we see a southern wind that is only changed with cold front passing through. Relatively flat topography in our coastal regions, so less than 25 feet, and prolonged and frequent rainfall events due to cold fronts and or in the summertime tropical weather. So the minimum number of samples needed to classify an area. To actually classify an area requires a minimum of 15 samples. But if you're starting from scratch or you have an area that has not any prior data, then a minimum of 30 samples is required. So we want to look at 3, 6, 9, and 12-year blocks of data. We need to know the median most probable number and know that it, for approved classification or approved status, that it is less than 14 MPN, and that we do not exceed 10% of the samples in the data set should not exceed 43 MPN. So to calculate median, the old days, staff would actually take graphing paper, rank the numbers, write it out, do the calculations on paper. And now, fortunately, I have Excel and we can plug in formulas. It's a lot easier. The calculation of percent greater than 43 is you count the number of times you exceed 43 and divide it by your total number of samples and then multiply it by 100. So here's a data set of Trinity Bay. We're looking at 9 years. And as you can see, circled in red are 3 sample stations that do exceed the 10% greater than 43 MPN. So these areas would not meet approved status criteria with the overall parameters. Therefore, they are classified as conditionally approved areas. So what are our classifications in the state of Texas? We utilize prohibited, restricted. We do not use conditionally restricted. We do use approved and conditionally approved. So prohibited areas, there's no harvest of shell stock for any purpose, except for depletion, gathering a seed stock, or nursery culture for aquaculture, all of which require Parks and Wildlife licensing. The prohibited classification is essentially the default for all unsurveyed areas. It's the classification for areas that are contaminated with biotoxins or chemical contaminants, and is also the classification for safety zones that we have immediately around wastewater treatment facilities, marinas, and fishing camps. Restricted areas, shellfish cannot be harvested or sold for direct marketing. The median MPN should not be greater than 88, and no more than 10% of the sample should exceed a 260 most probable number. Shellfish can be relayed from these areas to approved areas. That also requires a special permit from Texas Parks and Wildlife. The relaying process requires that the shell stock is moved to an area that has 14 consecutive days in the open status that allows the oysters to purge any potential bacteria or contaminants they have accumulated. Conditionally restricted areas, like I mentioned, we don't use them in Texas, and they're fairly rare in Gulf states. Approved areas, these oysters are essentially ready to eat right after harvest. They're relatively free of contaminants, biotoxins, and fecal coliform levels are not in dangerous concentrations. So they fall below the level of 14 MPN, and no more than 10% of the sample results have an MPN greater than 43. Conditionally approved areas, this is the most labor intensive classification. Areas meet approved criteria except for certain conditions. The certain conditions have to be predictable and definable. So it could be rainfall triggers or river elevations, river discharge volumes, something that we can quantitate and predict that it would negatively affect the growing area. They require a management plan. When the closure criteria is set in the management plan, it's not exceeded, the area can be in open status. When the criteria is exceeded, the area must be closed. The closure happens within typically 24 hours of the morning reporting. And like I said, it requires extra commitment or resources. You're potentially changing the status of these areas as rain or river runoff require. So this is a classification map that DSHS has generated for the Galveston Bay estuary. This has the 4 classifications we utilize. The blue is approved. The gray diagonal lines are conditionally approved. Prohibited are the small areas that are checkered or crosshatched. And then the dappled areas are the restricted areas. So conditionally managed areas, events causing the degraded water quality must be predictable and definable. So this table is also of 9 years of Trinity Bay data plots with specific trigger points. For example, less than 2.5 inches of rain at a specific rain gauge and or less than 20 feet river elevation are looked at a specific gauging station. Incremental rainfall and river stage totals are observed to see where the median exceeds the 14 or the percent greater than 43 exceeds 10%. And once we find those numbers, we're able to make essentially our closure criteria. So Texas growing area 2, 7-day cumulative rainfall is more than 2.5 inches at Anahuac, or the Trinity River that exceeds 20 feet at the Liberty gauge. It's the water quality studies that essentially help us to create these triggers. The field staff report the rainfall every morning, and then if we do have an exceedance, the Austin staff then issue a marine order to close a conditionally or approved area that has exceeded criteria. And the order goes into effect at 12:01 the following day. Texas Parks and Wildlife Game Wardens are the enforcement arm. And so they are typically afforded the 12 to 24 hour window of notice. So do we have any questions?
Andy, thank you so much for that presentation. I learned a lot and I'm intrigued. Now I wanted to maybe just kick off with a couple of observations and a question for you. I took note that you mentioned there are, I think it was annually, 2,300 base samples each year. And that you classify the results and you actually do some trends analysis. And you broke some of that information down for us. I was wondering, kind of looking through the non-point source lens that we focus on at Clean Coast, Texas, I was wondering if y'all ever try to track anything back to any particular land use or any data source other than which you kind of qualified under the adverse pollution condition criteria.
Absolutely, yeah. So that would fall into our shoreline surveys. In doing so, we look at, we're going to count livestock in a pasture, actual count heads that's recorded. We look to see if there are any obvious leach fields that have failed from houses that aren't on wastewater treatment plants. And that's looked at annually and more in depth at the triennial, and obviously more comprehensive at the comprehensive level. So we tend to track, and it's honestly, we are looking at the entire Gulf Coast as it relates to our specific harvest areas. So it's a huge undoing. Our field staff, they can conduct some of the shoreline surveys as they're running water samples. If they notice new houses going in or houses being added to a wastewater treatment facility, that's all tracked. If there's a new plant going in that would potentially impact an area, that's all tracked in our shoreline surveys.
Well, thank you for that, Andy. And sometimes a question like that is easier to ask than it is to actually find the needle in the haystack, or the needles in the haystacks, if you will, and trying to investigate coastal non-point source pollution or changes in water quality conditions.
Right.
We'd like to remind folks that if you do have a question, you can please submit it in the chat area and we will address that. I'd like to invite Brian to kick us off on the chat questions if we're ready to go with those.
Yeah, thank you, Jason. Yeah, we can start off. Kevin Frenzel asks, you're focusing on bacteria. Are there any concerns about inorganics such as mercury?
Oysters have been tested at the state level in some areas. Typically, mercury is not an issue. With the closure or the ban around the plant, the Alcoa plant, those oysters were actually tested, and they don't seem to bioaccumulate mercury specifically. So it's typically not an issue. And it likely could be that they're feeding at a much lower trophic level, whereas, a trout or something is feeding at a much higher level in the food web, and that bioamplification occurs at a much larger rate for those specific high-end predators.
Thank you for that. And kind of just quick expansion on that. Just, I'm curious, are there any other things that they are bioaccumulating that, you said they weren't specifically bioaccumulating mercury? What else might they be bioaccumulating?
Well, so we do track industrial plants and stuff that could potentially impact those oysters, or spill events or something like that. So our surveys surveys that relate to the classification tend to classify growing or harvest areas in a specific way to protect those that would actually hit the market. But there are some questions microplastics. There's a lot of ongoing studies. DSHS isn't currently conducting any, but some universities are looking at bioplastics, dioxins, PCBs, those kinds of contaminants.
All right. Yeah, thank you. Got another question from Mark. This is, what tools do you have at your disposal for anyone who is violating the rules?
Oh, yeah. So if we're doing water sampling, and it's happened before, a harvester's over a line or in an area that is either prohibited or it's in a closed status, we work hand in hand with Texas game wardens, and depending on the violation, they can be arrested, they can be fined. So the game wardens are very instrumental in helping us protect oyster consumers.
All right, I've got a couple other questions here. One's asking about your field staffing levels. Just like, what is the size of the staff that is out there conducting the surveys and the water samples?
Yeah, we're kind of a lean, mean crew. We have, so field staff also conducts the inspections on shucker packers, shippers. We all wear multiple hats. So we're looking at 7 field staff, and then we have 3, including myself, in Austin.
All right, so you guys are staying plenty busy. And is there any specific question regarding Copano Bay? Are there any plans to test for metals there, or is it something that might just be included in the routine sampling?
We don't routinely look at chemical contaminants. Basically, that portion of the program is highly dependent on funding, and there is no current plans to look at contaminants in Copano Bay. The actual analysis, collection of samples is relatively cheap, tissue analysis, but the laboratory analysis is substantially more expensive and it's pretty limiting. So typically we rely on grants specific to areas to analyze for issuing any kind of fish consumption advisories.
All right. Yeah, thanks for the clarification on that. I will give everybody another minute to type in any additional questions. I'm not seeing any more at the moment.
I'll jump
Okay.
in with one here, Andy, and kind of back to the trends analysis that you had mentioned earlier. And I don't know if I'm going to ask a trick question or not. So if just bear with me here. Have in your longer term analysis, have y'all identified any particular type of trend, improvements in water quality, or in areas where there might be issues or advisories, or kind of flatline, neither improvements or declines?
Right, that's a pretty complex question to unpack, because we do see trends that follow precipitation. So when we get into a drought, typically water quality improves. You have less runoff. In years where we get substantial rainfall, or we have a series of large events, tropical weather or just back-to-back cold fronts, we will see a degrading of water quality. The classification can be altered at the annual level, and so we do that to reflect those areas so if it's close to the trigger point, or if it's potentially needs to be reclassified, we can do so based off of even we typically look at 3-year blocks, so that we're not just acutely looking at changes, but we do have that longer data set and have a bigger picture. So atmospherically, the rainfall, that has a huge level of impact to growing areas. Development can alter things, but initial development homes without wastewater treatment, followed by then the institution of wastewater treatment, will typically, you'll see initial that potential degradation and then an improvement, because the wastewater treatment practices are much better than old leach field systems for individual homes.
Thanks for venturing into those deep waters with that question there, Andy. Looks like we do have one final question before we begin to scale down here, and I'll just go ahead and launch into this one. Is water salinity tested and recorded also?
Absolutely, yeah. So we do have thresholds to potentially reopen an area when an event has taken place. We will not reopen a harvest area if the salinity is below 2 parts per thousand. So salinity is recorded every time we dip the water. We take a bunch of other data points as well: weather conditions, is it cloudy, sunny, tide incoming, outgoing, wind speed, wind direction, anything that we could potentially quantify or analyze that may affect water quality as a trend, or contributing to the overall data set. But salinity is very important. Salinity is also important to oysters in that there's optimal feeding salinities. There's salinities that if they're below for a certain period of time, they simply perish because they can't feed, they can't maintain osmosis. So yeah, we track salinity with every single sample.
And I also have a follow-up to a little bit of guidance up to the Cope and Obey question asked earlier. You mentioned you guys don't regularly look for chemical contaminants. Is there a government organization responsible for regular analysis of chemical contaminants?
So DSHS, or through the Commissioner of Health, we are the agency that is tasked with doing so. But there is not regular funding allocated specific to marine or inland tissue analysis. So essentially, we would be the agency that can do it based on issuing consumption advice. There obviously are other entities that conduct these studies for academic purposes, but we are the only agency with regulatory authority.
All right. Yeah, thanks for diving a little bit deeper into that. And we did have the third one last question here pop up. David wants to know, with oyster farming, is it allowable to move 2-year oysters to a secondary farm site with higher salinity?
Typically, the oyster relaying, that falls under Texas Parks and Wildlife rules. So that's a little beyond my scope. As far as oysters have to be from an approved area if they are moved for our purposes for at least 14 days. So you could potentially, with approval from Parks and Wildlife or a permit, move them from a lower classification to a higher one.
All right. So David, yeah, if you want to know a little bit more, Texas Parks and Wildlife might be the way to go. We've had one more question pop in, and they want Mark is asking, can someone view the online results for the testing you guys do?
We do not publish the individual results. Obviously, that data is all subject to open records requests. However, we do maintain a status sheet for each harvest area. That is located on our website, and it's updated as changes are made. We also maintain a 1-800 number to update those status changes. And you can simply call our office to get some of that information over the phone.
All right, sounds great. I want to thank you for coming here today and explaining all of this to us. It's absolutely great information to know. And I want to pass it back to Jason for any final comments.
Yeah, Andy, I have to be honest, I'm pretty blown away with you kind of lifted the curtain a little bit on the depth of the work that you all are doing. And I kind of hope this opens up an opportunity for us to have some more engagement. And I'd like to learn more about your data and what y'all do, and to see maybe if we can point that in a little bit different direction for some other purposes, to see how that can collaborate and work with some of the other data that's being collected in the coastal zone.
Absolutely, yeah.
So thank you again very much for this great presentation, sharing your time and expertise, and sitting in the hot seat with us for about an hour. So thank you again.
Appreciate it.
So Andy's presentation wraps up a handful of presentations we've had here with Lunch and Learn. The first one looking at bacteria and water quality in the Gulf Coast. We looked at septic systems and assistance programs for improving their operations, also harmful algal blooms, as well as climate and weather changes. And then, of course, Andy's here focusing on our shellfish industry. And then next month, we have Dr. Wetz from Texas A&M Corpus Christi HRI, who will be presenting on water quality as well. After that, we're going to take a break for November and December, but we'll start a whole new series up starting in 2022. If you have any active, ongoing hot topics or research, feel free to reach out to us to see if we might be able to include those in a future Lunch and Learn series. And so we're excited about that. And I'd like to turn it back over to Adriana.
Thank you everyone for attending. I posted on the chat a few useful links. It tells you how you can get more information about our program. How can you view the previous Lunch and Learn, the recording for that. We had a question, Matt. I will share a link directly with you. And if you will be so kind to complete our last and final poll, so we can know whether you'd like to communicate with us, get information, and receive our emails. So we will greatly appreciate that. If you have not shared your email with us, please do so in the chat box so we can have that information. Okay, it seems like everyone has answered that. And if you would like to learn more about Clean Coast Texas, I'm just sharing the link for our website, the chat right now. And you can explore everything that we do. And I'll send links to our YouTube channel where you can find the recordings for all our talks since March. So I think we did great today. Thank you, everyone. Thank you, Andy. Thank you for attending. And this is a wrap up.
Thank you.
Take care
Bye-bye.
and be safe. Bye-bye.